Good evening, everyone. Today, I will talk to you about the biotechnological potential of one macroalga, especially regarding its application for pollution, biomonitoring, and remediation. I am Dr. Maria Areco, and with Dr. Dos Santos Afonso and Dr. Salomone, we have published this manuscript, and I am here to tell you a little bit more about it. Climate change is a complex phenomenon driven by a range of interconnected factors. The primary causes of climate change are the increased concentrations of greenhouse gases in the atmosphere. These gases include carbon dioxide, methane, and nitrous oxides, among others. The heat got trapped and created a greenhouse effect, leading to a rise in global temperatures. As you know, human activities are the main contributors to the heightened levels of greenhouse gases. Burning fossil fuels for energy and transportation releases substantial amounts of CO2 into the atmosphere. Deforestation and changes in land use also play a significant role, as trees and vegetation, and as we will see, algae act as natural carbon sinks absorbing CO2. Additionally, industrial processes, agriculture, and livestock production also release greenhouse gases. Changes in land use, urbanization, and waste management impact on the carbon cycle, further intensifying the climate crisis. But why am I talking to you about climate change if this talk is about the biotechnological potential of macroalgae? Well, because algae hold remarkable biotechnological potential to contribute significantly to climate change mitigation. These organisms have the unique ability to capture CO2 from the atmosphere during photosynthesis, and they act as natural carbon sinks, as trees. Algae can be cultivated in a wide range of environments, including wastewaters. They can capture industrial emissions, and in this way they help to reduce greenhouse emissions directly. Also, they are a source of lipids, pigments, vitamins, just to mention a few. The biotechnological capabilities of algae includes not also carbon reduction, but they present opportunities for sustainable resource utilization and the development of innovative, eco-friendly solutions for a more resilient future. Today, I will focus on the biotechnological potential of one particular macroalga, Ulva lactuca, also known as sea lettuce. I am sure many of you have seen this microalga at the beach or attached to the rocks at the shoreline. This alga has a widespread distribution and can be found along the coast shores around the world. Why I have chosen this particular macroalga? Because it possesses significant biotechnological potential with promising implications that may mitigate climate change. This macroalga rapid growth and the ability to absorb nutrients make it an ideal candidate for nutrient bioextraction from polluted waters, helping in the mitigation of eutrophication. Moreover, Ulva lactuca can serve as a valuable source for bioactive compounds with pharmaceutical and nutraceutical applications. Its high protein content also presents opportunities for sustainable aquaculture feed and bioenergy production through anaerobic digestion. Also, it can be used as a biofertilizer. It contributes to environmental remediation, resource optimization, and the development of innovative solutions for various industries. It is a profitable resource that can be used based on circular economy, in this way mitigating the impact on climate change in the process. Once again, climate change. They play a fundamental role in nutrient cycle of coastal system. They reflect changes in water quality in an efficient way. While growing, living microalgae can remove simultaneously different contaminants also, such as metals or pharmaceutical compounds. The excess of nutrients such as nitrate, phosphates, among others, from marine waters, and they can capture obviously CO2 emissions. Therefore, they might be used to reduce level of pollutants in aquaculture wastewaters. Macroalgae show great tolerance to metal contamination, and this is very important. For example, the metal retained in the cell wall can account for up to 95% of the total metal accumulated by the microalgae. Of course, this accumulation capacity of metals and other pollutants depends on numerous factors such as waves, exposure, temperature, salinity for the period, among other physicochemical aspects in the surroundings, and of course on the affinity of the species for each element or each pollutant. Since Ulva lactuca has resistance to high variation of salinity, it also has a high rate of nutrient assimilation, and it tolerates different pollutants under toxic conditions. It can be used as biomonitors, and here is the word. Biomonitor are organisms that contain information on the quantitative aspects of the quality of the environment. They provide a measure of the degree of the environmental contamination, so they are very important. For that, these organisms need to be sensitive to specific pollutant and tolerate high concentrations of them in the environment. They have to have the ability to accumulate this contaminant without being severely affected or killed by levels encountered in the environment. Otherwise, they cannot be used as biomonitors. They should be sessiles to be representative of the area of study, and they have to be abundant throughout most of the world coast, so we can compare, no? So biomonitors can provide a time-integrated measure of nutrient availability in coastal waters, providing qualitative information on the contamination level and environmental quality in seawaters. In this sense, Ulva lactuca has numerous features that make it one of the best bioindicators of metal pollutions in marine environments due to their sedentary lifestyle, considerable biomass, and easy identification and collection. So let's talk about Ulva lactuca remediation capacity. When there is an increase in nutrient concentration in the environment, there's a possibility to have eutrophication events. This leads to an excessive growth of plants, macroalgae, and microalgae, and have economics and environmental effects in coastal shores around the world. Ulva lactuca is an ideal candidate to analyze nutrient uptake kinetics and apply standardized function for an accurate analysis of nutrient uptake. Why? Because in general, Ulva lactuca has a maximum thickness of 2 cell layers, and I said in general because there are a few strains that doesn't have it. And then every cell is in contact with the environment. So at high concentrations of nitrate and phosphate, the rapid growth and higher photosynthesis of Ulva lactuca have been attributed to its high surface area volume ratios compared with other macroalgae. Furthermore, these macroalgae may reduce the occurrence of microalgae harmful blooms by reducing the nutrient available in water columns. Here you can see that Ulva lactuca is widely distributed. You can see that these authors publish studies with Ulva lactuca collected from Italy, Egypt, Algeria, India, Greece, Syria, and there are a lot of other papers from other parts of the world. These algae can accumulate essential and non-essential elements at different concentrations, and it showed different responses to toxic metals, of course, depending on the season, sampling period, age, the physicochemical condition of the sampling sites. This metal content in the biomass is significantly correlated with the levels of trace elements in water and sediments, but especially in waters. Ulva lactuca is a great accumulator of non-essential metals. We can see here examples of accumulation of cadmium, chromium, copper, nickel, lead, and zinc. So it could be used for biomonitoring and remediation of heavy metals also. Well, as you might be thinking, metal bioaccumulation and toxic effect in primary producers may have implication for higher trophic level, and it does, but we won't talk about this in this presentation. But what happened with other contaminants in the environments besides heavy metals or nutrients? What happened with persistent organic pollutants? I will refer them as POPs. POPs attract increasing attention for the widespread occurrence in the environment and their toxicity, mutagenic, and carcinogenic characteristic. Among them, we can mention antibiotics such as sulfatiazole, commonly used in aquaculture, diesel fuel production, growth factor, pesticides such as endosulfan, PCB, glyphosate, among many others. Well, in this image, you can see the sources of and fate of different POPs along the coastal zone. It was published by Merhabi and collaborators in 2020. I took it and changed it a little bit. And alga has a high capacity to be in POPs, so they might be considered as a suitable contaminant biomonitor. And why? Because the extent of POP bioaccumulation by alga is related to, of course, the characteristic of the organic substances, the macroalgae species, and different environmental factors, as I have previously mentioned, but also on the lipid content of the microalgae. Many studies have reported that nitrogen deficiency induced a significant increase in algal lipid content and enhanced the bioconcentration of POPs. Why? Because POPs are hydrophobic, then they tend to associate with lipids. So lipids are very important. An example of the application of Ulva as a biomonitor was given by Chini and collaborators in a paper published in 2019, where they found that the bioaccumulation of PCBs by seaweed bloom showed that PCB concentration were the highest near the contamination source and provide evidence about its possible role in the trophic transfers of PCB and other bioaccumulated pollutants. The fact that live macroalgae can simultaneously remove different pollutants is an interesting aspect because real waters are complex systems containing different ions that affect the removal capacity of a particular ion. So this alga can help in the remediation of polluted aquatic environments by absorbing and accumulating heavy metals and other contaminants. But how does alga remedy metals and organic pollutants? Well, some of the processes are metabolically active process by which the algae incorporate the contaminant. This is absorption with a B, and it's metabolically mediated. Others are passive process. It does not depend on the metabolic state of the organism, and it is adsorption with a D. This process is a reversible chemical reaction that takes place when an ion of a solution is exchanged for another of the same sign, as we can see here in the figure. And this ion is attached to a functional group, so changes in the ionic strength of the solution modify the adsorption process and other physicochemical characteristic of the media also. This process takes place continuously in nature, both in inorganic material as in living cells. The Ulva gender macroalgae contain 3 polysaccharides, starch, cellulose, and anionic compounds containing sulfate groups. Ulvan is a cell wall polysaccharide that contributes from 9 to 36% of the dry weight of the biomass of Ulva. They are highly sulfated and essential compounds of rhamnose 3-sulfate and other compounds. Here we can see this is one of the components of Ulva, rhamnose 3-sulfates, where adsorption on the cell wall take place. These images show the surface of Ulva lactuca through scanning electron microscopy at different magnification. Here is where adsorption takes place, and you can see here that you can see the two cell layer of Ulva lactuca. Here is an example where we have demonstrated the capacity of Ulva lactuca to absorb glyphosate. Results demonstrate that this binding is mediated by copper. But let's talk about the formation of sulfate complex between biomass and metal ions. This formation involves the coordination of oxygen donor atoms that constitute alga cell walls, followed by the release of proton from the surface, as we have seen. Theoretically, in this example, copper is a free metal ion in solution. This is the sorbent surface with site which is susceptible for the coordination. So here is the surface complex formed on the sorbent. Also, surface complex can be formed between some biomass cell walls coordination groups and dissolvable ligands such as glyphosate, following in this case the release of water or hydroxyl from the surface. And certainly ternary complexes formation involving simultaneously or consecutive absorption of metal ions and ligand is possible. But results demonstrate that PMG or glyphosate adsorption only happens in the presence of copper. And also that copper adsorption is higher when there is presence of PMG or glyphosate, compared with copper adsorption when there's no glyphosate in the media. These images demonstrate the presence of heavy metals on the surface of the algae after being exposed to different heavy metals such as cadmium and copper, zinc, lead, and of course nickel here in the mapping images. So Ulva can be used for the remediation of different pollutants, as you can see in this table where the results of different authors are shown. But today most of the naturally produced and harvested biomass of Ulva lactuca is an unused resource. Then a huge amount of biomass is generated and available for alternative purposes such as a source of pigments, lipids, and adsorbent of materials for aquaculture or agricultural application, and for cosmetic and pharmaceutical uses. So I will talk to you the last minute about this biotechnological potential. It can be cultivated in an integrated multi-trophic aquaculture system. It can use nutrients released by fish or shrimp farming, reducing environmental impacts and providing additionally economical values to the process. Its nutrient-rich biomass can be processed and used as organic fertilizer for agricultural purposes. Its high nitrogen and potassium content make it a valuable soil amendment. Also, it can be used for nutritional supplements because it's rich in vitamins, minerals, and proteins, making it a potential source of nutritional supplement for both human and animals. Also for biogas production, since its biomass can be used in an aerobic digestion process to produce biogas, which can then be used for energy generation, and many other uses such as animal feed, wastewater treatment, as I talked before. In conclusion, Microalgae-based technology may be a viable, cost-effective, and a greener option to reduce the rejection of priority hazardous substances in contaminated waters. Furthermore, they can be applied for the development of algae-based products and technology and to the design of algae biorefinery. These kind of developments are needed in order to minimize the effects of anthropogenic activities in the environment while reducing our carbon footprint. Climate change is a complex phenomenon driven by a range of interconnected factors, as we just see. And our goal is to keep on studying the biotechnological potential of algae and the development of new algae-based technology to minimize the impact of climate change. So thank you very, very much for your attention.